Over nearly 3 decades, our understanding of depression has evolved from a simple chemical imbalance model to a dynamic perspective of neural circuits, synaptic strength, and the molecular cascades governing plasticity. Kandel argued in 1998 for a common biological framework reuniting psychiatry with medicine, establishing the foundational principle that experience alters the strength of synaptic connections. Depression is now viewed as these dynamic plasticity mechanisms driven in the wrong direction by stress and genetic vulnerability, exhausting the brain's capacity for adaptive remodeling. This text synthesizes the current neurobiological evidence for this molecular and circuit perspective. The initial monoamine era accidentally discovered drugs like reserpine depleted monoamines and precipitated depression, while iproniazid elevated mood, leading to the hypotheses focusing on norepinephrine and serotonin that were seemingly confirmed by the success of SSRIs. However, this framework was always incomplete, as the temporal dissociation between rapid serotonin transporter blockade and the weeks required for clinical improvement demonstrated that the initial pharmacological action is not the therapeutic mechanism. The therapeutic mechanism lies downstream in adaptive changes in receptor sensitivity, gene transcription, and ultimately synaptic remodeling, where monoamines function as modulators rather than the final common pathway. Depression is fundamentally a disorder of distributed neural circuits, where functional neuroimaging consistently identifies a pattern of hyperactivity in limbic regions, such as the subgenual anterior cingulate and amygdala, coupled with hypoactivity in dorsal prefrontal and parietal regulatory regions. Medial prefrontal and posterior cingulate nodes within the default mode network show pathological hyperconnectivity underlying ruminative processing. Deep brain stimulation of Area 25 provided critical proof of principle that modulating defined nodes can reverse a pathological state. These circuit abnormalities reflect failures of single-cell experience-dependent remodeling. Shift in focus from neurotransmitters to intracellular signaling and structural plasticity led to the neurotrophic and synaptic plasticity hypothesis. Chronic stress and depression reduce expression of brain-derived neurotrophic factor, or BDNF, in the hippocampus and prefrontal cortex, resulting in dendritic atrophy, spine loss, and ultimately neuronal dysfunction. Antidepressants converge on upregulating BDNF and activating the CREB signaling cascade to restore these excitatory synapses, which are defined as the fundamental lesion of the disorder. Integrated models include the concept of allostatic load as the bridge between psychosocial stress and molecular pathology. In depression, chronic hypothalamic pituitary adrenal axis activation causes cortisol non-suppression, indicating impaired negative feedback loop. Excess glucocorticoids are directly neurotoxic to the hippocampus, impairing long-term potentiation and reducing neurogenesis. Stress disrupts glutamate homeostasis, promoting excitotoxic synaptic stress. Restoring adult hippocampal neurogenesis in the dentate gyrus appears mechanistically necessary for at least some antidepressant therapeutic actions. Single most important development in the 21st century is the glutamatergic revolution, which demonstrated rapid antidepressant effects through NMDA receptor antagonism. Low doses of ketamine produce clinical responses within hours, a satisfying sequence that compresses activity-dependent structural synaptic change from weeks into hours, depending on mTOR signaling and increased synaptic protein synthesis. Neuroinflammation serves as a compelling interface where peripheral cytokines activate microglia to induce sickness behavior syndromes overlapping with depression. GWS studies abandon search for single depression genes in favor of highly polygenic vulnerability distributed across thousands of variants of tiny individual effect. Resurgence of psilocybin research demonstrates rapid and sustained effects mediated by 5-HT2A receptor agonism on cortical neurons, producing transient heightened plasticity to reopen a critical period that allows entrenched maladaptive circuit patterns to be reorganized. Integrated synthesis supports model that genetic polygenic vulnerability encounters environmental stress, overwhelming homeostatic plasticity and creating self-reinforcing pathological state characterized by HPA hyperactivity, reduced BDNF signaling, microglial activation, disrupted glutamate homeostasis, and maladaptive remodeling of circuits. Effective treatments including SSRIs acting over weeks, ketamine over hours, psychotherapy over months, and psilocybin over days all converge on the restoration of synaptic connectivity. Monoamines modulators, synapse locus. Disorder mind is disordered brain. Disordered brain can be repaired.